The effect of ammonium and potassium nitrate on Metellus edulis' oxygen and ATP deficiency and on the PKLR gene expression by Christine Yu. Wastewater production is constantly increasing globally. In 1960, the amount of wastewater production was under 1,000 cubic kilometers, but by 2000, the water pollution rose to above 10,000 cubic kilometers. At this rate, by 2060, wastewater production will triple in comparison to 1960. Water pollutants, including nitrogen, usually come from agricultural products, urban pollutants, industrial pollutants, and water treatment pollutants. Nitrogen is often found in fertilizers, which makes the consumption rate relatively high. Though nitrogen is good for our ecosystem, an excessive amount will negatively damage the health of infants and adults. All the nitrogen waste products end up in fresh and marine waters. In the United States, nitrogen wastes in freshwater have significantly increased, with a 70% increase in most freshwater lakes and streams. The standard nitrate concentration is just at 10 milligrams per liter, but 52% of freshwater systems have a higher concentration. Nitrogen is a common element in nature. Approximately 78% of the Earth's atmosphere consists of nitrogen gas. As nitrogen naturally cycles through the air, soil, and water, it undergoes various chemical and biological transformations. These reactions result in the formation of nitrogen-based compounds and molecules, which are essential for the growth of plants, animals, and humans. Agricultural production is dependent on the cycling of nitrogen within the rural environment. Though nitrogen is essential, too much in a person's diet may cause nitrate poisoning. Animals with high nitrate levels in their diets accumulate nitrite. Nitrite is absorbed into the blood, which carries a reduction in the ability of the blood to carry oxygen from the lungs to the body tissues. When the blood can no longer supply oxygen to the body, the animal can suffocate. Mussels are important in the environment for many reasons. Mussels play a key role in aquatic environments because they modify aquatic habitat, making it more suitable for themselves and other organisms to live in. The mussels clean the water they live in by removing phytoplankton and the bacteria and fungi that are attached to the non-living organic particles they have removed from the water column. Metellus edulis is a blue mussel that lives in freshwater aquatic environments. Because of its habitat, the mussel would be an ideal organism to use because of the natural exposure to the pesticides in the water. The cerebral ganglia is a bunch of nerve cells found in the bivalves. It functions as a control center for the mussel. By dissecting the mussel and isolating the ganglia, the nitrate can be exposed to the cells, and the gene expression of the cells could be examined. The PKLR gene is expressed in the liver and in red blood cells. The red blood cells are carriers for oxygen. The gene is also involved in glycolysis, which aids in the production of ATP. ATP is the cell's main energy source. Therefore, this gene will appear if there is a deficiency of oxygen or ATP. In 2012, Jeremy Brill performed a study assessing the effects of native freshwater mussels on aquatic nitrogen dynamics. The goal of the study was to control the mussel's habitat and see the effects of the mussels on nitrogen dynamics. The variables included ammonium nitrate, organic nitrogen, nitrite, and total nitrogen. The effects were analyzed with a sensitivity analysis, which shows how an independent variable impacts a dependent variable. The results showed a deficiency in proper nutrients and oxygen in the mussels. In 2007, Claire McGuigan performed the study, The Effect of Nitrate on Human Cell Lines in Culture. The purpose of the study was to examine the effects of environmentally relevant concentrations of nitrate on cytotoxicity and protein expression in human cell lines. The liver and kidney cell lines were exposed to a nitrate salt for 24, 48, or 72 hours, and then examined for the effects on development or growth. Nitrate was cytotoxic to both cell types at high concentrations, but ammonium nitrate was generally more toxic than potassium nitrate. The liver and kidney cells displayed similar responses for most assays, but the protein expression did not change significantly. The purpose of the study is to test the gene expression of the PKLR gene, which expresses the amount of ATP and oxygen being produced in that organism when the Metellus edulis' nerve cells are exposed to the ammonium and potassium nitrate. The null hypothesis states that ammonium and potassium nitrate will have no effect on the gene expression of the ganglion nerve cells of Metellus edulis. The PKLR gene will not appear under the exposure of nitrate, and no abnormalities will occur in the development of the nerve cells. The alternate hypothesis states that ammonium and potassium nitrate will negatively impact and alter the gene expression of the ganglion nerve cells of Metellus edulis in that the PKLR gene will appear and be present. The higher the concentration of ammonium and potassium nitrate, the more abnormalities in gene expression and cell structure will occur. To perform the study, there will be a total of 50 muscles. Each will go under muscle dissection, and the ganglia will be extracted. If necessary, the ganglia will be stored at -70 degrees Celsius. After, the cells will be divided into 5 different groups. One control group that consists of 10 different cells, 2 1.0 milligrams per liter of ammonium nitrate and potassium nitrate, and 2 groups of 20 milligrams per liter of ammonium nitrate and potassium nitrate. The ganglia will be exposed to the appropriate concentration of nitrate for 24 hours. Then, it will undergo the performance of RNA isolation, RT-PCR, and gel electrophoresis. The gel electrophoresis will determine whether the PKLR gene will be expressed under the nitrate conditions. The size of the band, shown in gel electrophoresis, will be analyzed using the Western blot analysis, which detects the specific proteins with the use of gel electrophoresis. The size approximations are taken by comparing the stained bands to that of the marker or ladder loaded during the electrophoresis. A statistical analysis will be done using post-hoc Scheffé one-way ANOVA with a significance level less than 0.05. This slide shows the budget for this experiment. Many of the equipment that is necessary in this project is already supplied at the Manhasset Science Research Lab, so this project will be doable at this lab. This is my bibliography. Thank you for watching, and do you have any questions?